Enzymes discovered in gut bacteria can change a donor's blood group. When a life hangs in the balance, blood transfusions can help sustain a patient, but only if the donor's blood type is a match. A new discovery by researchers from Denmark and Sweden could help in those emergency situations while also easing global shortages in blood supplies. The team identified a mixture of enzymes made by a species of bacteria found in our guts that can, in lab studies, transform red blood cells into the universal type O with remarkable high efficiencies, improving on an idea hatched 40 years ago. Like virtually all cells in the human body, red blood cells are covered in a fuzz of unique sugary structures. These vary from person to person, with some sporting type A structures and others type B. Some have both A and B, and others have neither, which is designated as O. Immune systems that have never seen types A or B will attack and destroy those cells on sight if they receive them in a transfusion, whereas type O blood is far more widely accepted for most recipients. Because of this versatility, type O blood stocks are often used up, especially in medical emergencies, where doctors must act quickly without knowing a patient's blood type. Converting red blood cells into the universal type O is not a new idea. The technique was pioneered back in 1982 when scientists discovered an enzyme extracted from coffee beans that could strip type B cells of their surface sugars. But that enzyme reaction was very inefficient, making large-scale use impractical, and despite early promise in clinical trials, safety concerns were raised. For unknown reasons, donor blood was sometimes still incompatible in recipients despite donor cells being stripped of nearly all their antigens. So scientists went back to the drawing board, discovering other enzymes in collection of gut bacteria as recently as the year 2019. What's tricky is that there are now, as of 2022, more than 40 blood group systems beyond the ABO system and rhesus factor most are familiar with. Even with the A and B blood groups, there are subtypes with varying lengths and densities of the signature molecule protruding from the red cell's membranes. Here we report the discovery of remarkably efficient enzymes not only against A and B antigens, but also against their extensions. Matthias Jensen and Lynn Stenfeld, two bioengineers at the Technical University of Denmark, write in their published paper, along with their Swedish colleagues. Based on previous work by other teams, the researchers selected a few candidate enzymes made by the gut bacteria Akkermansia muciniphila and treated red blood cells from multiple donors and various A and B subtypes with them. Importantly, for potential clinical use, the enzymes were incubated with high concentrations of red blood cells at room temperature and for only 30 minutes, improving on the longer processing and less efficient conditions of previous candidates. These mild conditions without additives, for example, dextran, together with excellent enzyme efficacies, are important feasibility parameters in clinical applications, Jensen, Stenfeld, and colleagues note. Crucially, the chosen enzymes also remove all four known extensions of group A and B antigens from red blood cells, in addition to the shorter canonical A and B antigens of other blood subsystems. Removing the long sugary extensions reduced the incompatibility of treated B-type cells with plasma samples to less than 9% and made reactions less severe where they did happen. More work is required to understand why a small fraction of apparently sugarless red blood cells still cross-react with group O plasmas and to improve the conversion of group A blood cells. However, in finding enzymes that remove a greater variety of A and B antigens, the researchers say their studies uncovers a missing link in the production of universal blood for transfusion and potentially organs for transplantation. In 2022, researchers used a similar strategy with different enzymes to convert donated lungs from Group A blood type to the universal type O under lab conditions. This new work might improve those efforts enough to satisfy the safety standards required for human transplant trials. Lab-grown red blood cells are also in human trials to test whether they last longer than donated blood. If so, that could reduce the demand on blood supplies and also help patients requiring repeat transfusions avoid complications. The study was published in Nature Microbiology, and this is on Science Alert by Claire Watson. Please leave your comments. Thank you for your support. Kindly support my Patreon account. The daily posts are 5 videos daily, and they are totally different from what I have on my YouTube channel. Thank you so much for your support and that you find all my content so interesting. You'll find the Patreon account details in the description box below.